In the second part of this video, we'll look at some of the terms that are not covered within the National 5 course. We've discussed that autosomal conditions are ones which are on chromosomes 1 through 22, so the inheritance for those autosomal recessive and autosomal dominant are just like what we would have talked about in National 5 with recessive and dominant conditions. Incomplete dominance is quite a simple concept, but we'll come on to that. And sex-linked recessive is again simple as long as you remember to put your X's and Y's in. Autosomal recessive is just National 5 biology. So autosomal recessive conditions are expressed rarely in nature. What is required for an autosomal recessive condition is for the person affected to have 2 recessive alleles. This requires 2 humans to mate and produce offspring, not knowing that each has this recessive allele in their genotype. So it's very rare that it comes up together. These conditions can skip generations because someone can be what's called carrier. Male and females are equally affected because it's on the autosomal chromosomes, not the X and Y sex chromosomes. All sufferers are homozygous recessive, so they have 2 small letters. And non-sufferers can be either homozygous dominant or the heterozygote. An example of this would be cystic fibrosis. We can see here that this affected female mates with an unaffected male. It doesn't matter if they're males or females because we're looking at autosomal, but just for the terms. The mother and the father have 3 children, all of whom are unaffected. We don't know if the father has given his, dominant unaffected allele to these children here. It's very possible that he could be a heterozygote and who could be carrying one of these deleterious alleles. The female passes on, because it's a recessive condition, she has to be a homozygote. She passes on this to both her sons and her daughters, but in all cases, father passes on the dominant allele, so it overrides it. So we have phenotypically fine children. This son mates with a female who also is phenotypically fine. They have 2 phenotypically fine daughters, and the phenotypically fine daughter, number 1, mates with a male, and somehow they have 2 affected children, a son and a daughter. Now, both the female and the male here must be heterozygotes with one of the affected recessive characteristics as one of their alleles. So this can skip generations, and you don't see it for 2 or 3 or 4 generations, and then it pops up as soon as you mate with another person who's a heterozygote. In this one, we can actually use what we know about genetics to work it out, so you can pause the video and have a go working out who will be small c, small c, small c, or big C, or big C, small c. Okay, so the first thing you can do is you can go around all the affected people and put little c underneath them, because to be affected with the recessive characteristic that's autosomal, you have to be a homozygote. We can work out that because these 2 are able to produce an affected person, but they themselves are not affected, they must be heterozygotes. We don't know the genotype of these 2, we only know the phenotype is unaffected. We know these 2 similarly are heterozygotes, so I've got the dominant from one parent and the recessive from the other. So obviously a different family, anyway, because they are able to create a affected child. Similarly, affected and unaffected, because their phenotype is normal, they have a dominant allele, but the father can only pass on the recessive allele, so they will all be heterozygotes. Autosomal dominant is just dominant inheritance from National 5, so it appears every generation. Every sufferer that you see has to have a phenotypically affected parent, so you have to be able to see it in every generation. If you have children who do not express the trait in the phenotype, their children will not have the disorder. You can only have children with the disorder if one of the parents has the disorder. Again, because it's autosomal, males and females are equally affected, and all sufferers, all non-sufferers would be homozygous recessive, because a single dominant allele is enough to give you the disorder. An example would be Huntington's chorea. The inheritance of Huntington's is slightly more complicated and very interesting, but we won't go into that. You can see here an affected parent has an affected child, they have an affected child, and they pass on to their children. And you can work out the genotypes coming down here. Autosomal incomplete dominance is very interesting. It's using a non-human analogy. If you have a red flower, which is capital R, you mate it with a white flower, which is capital W. Their progeny will have a red allele and a white allele. Now, neither is dominant. You will get a pink flower. So that is an intermediate phenotype of the two parents. So similar to this, in the human example, the heterozygote exhibits a phenotype different from both of the homozygote parents. So in something like sickle cell anemia, the fully expressed form of the disorder is quite rare because usually these disorders are quite harmful. A partially expressed form occurs more frequently, so that is the heterozygote, and males and females, because it's autosomal, are equally affected. Non-sufferers, who are homozygous, are capital H in this example. So they have no traits of the deleterious allele, the bad one. Ones who are homozygous for the deleterious allele will have the disorder, and ones who have one of each of the alleles will be a heterozygote. They will have some intermediate phenotype. Interestingly, in terms of sickle cell anemia, you, for the most part, are likely to be capital H, having normal red blood cells, but being able to catch malaria. Interestingly, sickle cell anemia, which is capital H, prevents you from getting malaria, but gives you a horrible disorder where running or walking quickly will put you in agony because of your weirdly shaped red blood cells. Capital H, capital H, capital S, which is called sickle cell trait rather than sickle cell anemia. The red blood cells are oddly shaped, so it's difficult for the person to get malaria because the plasmodium can't get in, but they also don't have sickle cell anemia. So that is the best of both conditions. Sex-linked recessive is the last and slightly more complicated example. So the first thing to do is to know that females are XX and males are natally XY. What we'll do is we'll always put in the XX and the XY as the first thing, and then you would work out the genetics. That's the easiest way to not make a mistake. A lot of people in the prelims put in the H's, but forget to put in the X's and the Y's. So put in the X and Y's first. Lots more males are affected than females. That's because males only have one X chromosome. Sex-linked recessive conditions in the higher course are carried on the X chromosome. So if males have a negative recessive allele, that's the only X allele they have. So they will have the condition. Females can have an affected allele, but also have a good allele. So it's like autosomal recessive for them. Whereas for males, it's basically autosomal dominant all the time. None of the sons of an affected male will show the trait. That's because men are the sex determiners. Women will always pass an X chromosome onto their children. Men will either pass an X chromosome onto their children to make a female or a Y to make a male. If they pass their Y, that has nothing to do with the condition, so men cannot pass on these sex-linked conditions. Non-sufferers are homozygous dominant, so for men that is X with a normal allele and the Y, and for females that's X with the two X's with the normal ones. Women can be heterozygous and they can be what's called carriers. So for example, I have colour blindness, my mother is a carrier, and my dad gave me a Y chromosome. My mum gave me the dodgy X chromosome, so I am red-green colourblind. Had she given me the good X chromosome, I'd be fine. So this is another example. Colourblindness is one, haemophilia is another. It tends to get a bit busy underneath the letters here, so we put in circles are females, squares are males, so we do XY for all the boxes and XX for all the circles. This individual could, by its phenotype, either be a carrier, so X with a big H, X with a small h, or phenotypically and genotypically normal, so X with a big H, X with a big H. When they form a male, the male passes on his Y, and we can see in this example the female must have passed on a normal X rather than the possible carrier one here, because the male is fine. They make a daughter, however, who is a carrier, so the male passed on his negative X, his bad X, and the female passed on her good X. When this carrier female mates with this normal male, they produce one, so the man passes Y chromosome on, the female unfortunately passes on her affected X, so you get an affected male. They have lots of kids, so they have one of everything basically. So they have an affected X male, an unaffected X female male. They have a female here who we don't really know if she is a carrier or not. All we know is that she has at least one good X. And we have Hamish, who is X with a good H and Y. Here we have a carrier female. So as I said, this is the example of me. So my mother is X with a good H and X with a bad H. My dad is not colorblind. He gave me his Y chromosome, and my mother gave me the bad H chromosome. We quickly go through some examples. So Huntington's chorea is caused by a single dominant gene, and it's not sex-linked. So you now immediately decide that you can forget all that stuff about X's and Y's. A woman's father is heterozygous. So we're now picturing a big letter. So for example, big H and small h. And her mother was unaffected. So we're now picturing beside the big H and small h, two small h's. What are the chances that that woman got whatever the condition is? Now, if the condition is dominant, it says here, 50% of the time the dad would pass on his dominant bad allele, and 50% of the time he would pass on his good allele. The female is recessive, so homozygous recessive, true breeding, so she will pass on her small h's each time. So she is completely irrelevant for this. Anytime you have a cross between one individual and a homozygous recessive, you can just completely rule that person out if it's autosomal. So if the male passes on his affected chromosome, you will get the condition. If he passes on his unaffected, you won't, so it's 50-50. This one here, again, so it's sex-linked, so we immediately go individual 3 is a male, we'll put an XY in. Individual 4 is a female, put XX. Individual 3 has the condition, because males only get one shot, he must have the dodgy X chromosome. So beside his X, we put a small h. They haven't given you letters, I'm just following the H. So X with a small h and his Y. The individual 4, she's unaffected, but she's capable of making a male who is affected. So remember, this one here, even this is tricky because he is affected, so you would assume he's the one who's caused the affliction in his son. But actually, remember, men pass on a Y, so it's not his fault this time. So he's passed on the Y to make a male, and this lady here has passed on an affected X. So she must have, one of her X's must be the little h, it must be the recessive bad allele. She is white herself though, so she doesn't have the condition, so she must have a good big H, so X with a big H, and then an X with a small h. How come individual 1 can't pass the condition to his son? So individual 1 is affected, we've talked about this. Men cannot pass sex-linked recessive conditions to their progeny, their sons even, they can't to their daughters, because they pass the Y chromosome to their son, which does not contain that gene. This is one of my favourite ones because it takes about 20 minutes to work it out if you do it properly, or 3 seconds if you know how to cheat. So they want you to, just from this information, work out if the disorder is sex-linked or autosomal recessive or dominant. Only in multiple choice, only one of the answers can be correct. So the first thing I would do is try and see if it can be, always try and find out if it's 2 heterozygotes who have passed on a recessive allele. So in this occasion, could it be autosomal recessive? So could it be a heterozygote and heterozygote mating and 1 in 4 times making this affected person? Anytime 2 individuals make an individual who is not like them, autosomal recessive is what you're thinking. Sickle cell, autosomal blood disorder, which this is the last one, faulty form of the hemoglobin protein is made. The protein is inefficient at carrying oxygen. That's why they can't do a lot of exercise. The allele for normal is capital H. It's incomplete dominant to the affected allele S. Heterozygotes HS suffer from a milder version called sickle cell trait. The pedigree is shown below for 3 generations. Parental F1, F2. What is the genotype of individual 5? So they are a male, doesn't really matter though, and they have sickle cell disease. So they must have both of the S's, big S and big S, because they would be called sickle cell trait if they were a grey box, and they be unaffected if they were white. So it's big S, big S. 3 and 4 go on to have another child. What are the chances that they have the same genotype as their parents? So both 3 and 4 are sickle cell traits, so that means they are heterozygotes. When heterozygotes mate, both of them can give the H, or both of them can give the S. That will happen 1 in 4 times, that they both happen to pick the same letter. The other 2 and 4 times, one of them will pick an H, the other will pick an S. On those occasions, you will get a child who has the same genotype as their parent. So the answer is 50% of the time. Hopefully that made sense. You can see why I've split it across 2 videos. The first one mainly going through some of the National 5 revision, and the second one going through higher human. Hopefully that makes sense. Please do ask if it doesn't.